Hi everyone, we are MP007 from the Amati Group, and our project is on the development of lipopeptides as a combinatorial treatment for colorectal tumors. My name is Aryan.
My name is Bhargav.
I'm Manya.
And I'm Sarah.
So first, like a brief table of contents. We'll start with the introduction to our main condition that we're working with, which is colorectal cancer. Then we'll go into the treatment that we're developing, which is lipopeptides. Then we'll go into the cell line we are maintaining to test for lipopeptides, which is the HCT116 cell line. And lastly, we'll go over current progress and future plans. So first, a brief overview on colorectal cancer. Colorectal cancer is a very important and common cancer worldwide, usually affecting elderly people over the age of 50. According to the World Health Organization, colorectal cancer accounts for 10% of all cancer cases worldwide, and it is the second leading cause of cancer-related deaths in the world. This cancer starts in the colon or rectum in large intestine, as shown in the image on the top right. The cancer starts as benign colon polyps that don't heavily affect the patient at first, as shown on the bottom right image, but these polyps will grow bigger and eventually develop into cancerous cells and into a tumor if given enough time to grow. Once the cancer develops from the polyps, symptoms may include changes in bowel habits such as diarrhea or constipation, but blood and stool and abdominal pain can also occur. The cancer does lead to death often if not treated. So there are many current therapeutic measures used in the medical field today to first of all test for the cancer, but also to treat the cancer. Screening tests include colonoscopies and stool tests to detect the cancer in its early stages when it is the most treatable. Colonoscopies are, as shown in the image on the right, involve inserting a long and flexible tube with a live feed of a camera through the large intestine of a patient to physically detect the presence of any polyps or cancer tumors. And stool tests can also be used to test for symptoms of the cancer. So now into treatments. Treatment options for the cancer largely depend on the stage of the cancer, such as if it's in its early stages when it's easily testable and it's treatable, or late stages. Common options include surgery or immunotherapy, but the main problem with these treatments and tests are that they are often invasive for the patient, such as using surgery to forcefully remove this polyp or tumor from the intestine. And to add on to that, these treatments are mostly applicable to patients in earlier stages where the tumor can be easily removed. But many of these treatments will not work with patients in later stages of the cancer when the cancer has spread to their whole intestine. The main purpose of our project is to develop a non-invasive therapeutic technique involving lipopeptides to treat colorectal tumors.
So next we're going to talk about lipopeptides, which are the method that we're going to use to kill the colorectal cancer tumors. And so a lipopeptide is a series of amino acids which are conjugated to a lipid molecule. And lipopeptides have many properties that make them beneficial to helping kill cancer cells. They are both antimicrobial and cytotoxic, which make them widely used in the food industry. And they have also been proven that they can trigger programmed cell death or apoptosis in cancer cells. They do this by activating apoptotic pathways or inhibiting anti-apoptotic pathways within the cells. They also tend to inhibit angiogenesis, or the process of new blood vessel formation in cancer tumors, and this is essential for tumor growth and metastasis. Some lipopeptides have been shown to inhibit angiogenesis, thereby limiting the blood supply to tumors and hindering their growth. It has also been proven that lipopeptides target cancer cells and that certain types of lipopeptides can selectively target cancer cells over healthy cells. This selectivity can be based on differences in membrane composition, surface receptors, or other molecular characteristics unique to cancer cells. Lipopeptides can also modulate the immune response against cancer cells. They may stimulate immune cells such as T cells and natural killer cells to recognize and attack cancer cells more effectively. They also interfere with various signaling pathways that are dysregulated in cancer cells. By targeting key proteins involved in cell proliferation, survival, and metastasis, lipopeptides can disrupt cancer-promoting signals and inhibit tumor growth. They also serve as carriers for anti-cancer drugs or imaging agents, allowing targeted delivery to cancer cells. This targeted delivery can improve the efficacy of conventional cancer treatments while minimizing side effects on healthy tissues. They also hinder cell cancer's proliferation due to the presence of free COOH, which causes an interaction between lipopeptides and the cancer cell membrane. Fengisin is known to be the most effective lipopeptide for colorectal cancer cell proliferation, though others like surfactant and niturin are also successful in doing this. So now we're going to talk about our procedure to create lipopeptides. So you start by attaching the amino acid to a lipid, and you're going to wash the amino acid to get rid of excess byproducts. The next step is deprotection, which involves the removal of an N-alpha group using either TMSI or zinc bromide in order to expose the amino group of the amino acid for the coupling reaction. You're going to wash this again to get rid of any byproducts, and then you're going to couple the next amino acid, which will form a peptide bond. You're going to filter and wash this again to remove excess reagents. You're going to keep on repeating this cycle for the desired lipopeptide. Then all protecting groups on the lipopeptide will be removed to expose the amino and carboxyl groups of the lipopeptide. The peptide is then cleaved from the resin and washed one more time, either in a column or using a type of liquid chromatography called a preparative LC.
So next we're going to be discussing the cells that we used in our research. To conduct our research, we used the HCT116 cell line, which is a human colorectal carcinoma cell line, which is derived from adult males. And the cell line is very commonly used, and it's very useful in conducting assays such as apoptosis assays, and you can use that to determine the effects of different molecules on cancer cells. These cells tend to double around every 18 hours, and they reach full confluency or their highest concentration every 2 to 3 days. And these cells, healthy cells, appear as small and circular under the microscope. In order to maintain these cells, we prepare a working media, which includes 45 milliliters of McCoy's 5A, which is a general media, 4.5 milliliters of FBS, which is fetal bovine serum, and this provides the cells with the nutrients that they need to grow, and 0.5 ml of penstrep, which is to prevent contamination and bacteria. So some of the challenges that we've had with our cell line are primarily the contamination of cells, especially when we first started. This was mainly caused by our technique. For example, when we would transport cells, the cells might touch the cap of the flask, which would expose them to different contaminants. And also, since a lot of our workspaces are shared with other groups and other cells, this could lead to cross-contamination between flasks. For example, in our cell culture hoods or in our water baths, which are not properly cleaned and maintained. Another big problem that we've had was a really slow cell growth. So like we talked about how cells are supposed to reach confluency every 2 to 3 days. Initially, we had cells reaching confluency only about every 5 days. And this was primarily due to an inconsistent media. Initially, our media was lacking FBS and we had a lower concentration, and we adjusted our procedure to include a higher concentration of FBS to allow them to grow faster. Another problem was also low CO2 conditions in one of the incubators that we were using to grow our cells. And over-trypsinization of cells also led to a slow growth and the death of many cells. So just to go over trypsinization, HCT116 cells are what we call an adherent cell line, which means that they grow attached to the flask. So in order to split the cells and to create new flasks of cells, we would need to first detach them from the flasks and then split them. And the process of detachment uses trypsin. And when we use trypsin, we need to incubate the cells for a certain period of time to ensure that they're detached. So our initial procedure had a certain trypsinization time, which might have been excessive, and that could cause a lot of the cells to die during our procedure. And these images, the top image shows a healthy HCT116 cell flask. And as you can see, the cells are really clumped together and they cover the entire flask. And in the bottom picture, you can see dead cells, which are not completely attached and just floating around in the media. So some of the solutions that we came up with was first, to prevent contamination, we cleared out the incubator and hood, and we cleaned them. So we sprayed them with ethanol and with a special spray to prevent mycoplasma contamination. And then we also prepared new working media. So we discarded the old one because we suspected that it might be contaminated. And then we also increased the ratio of media to cells in our flasks. We initially started with 4 milliliters of media for each flask, and we increased that to 6 milliliters in order to provide more nutrients for the cells to grow. Then we also decreased the incubation time post-trypsinization to avoid over-trypsinization. And then we also switched the general media that we used in our working media. So we started with McCoy's 5A media, which is a certain type, and then we changed it to DMEM, which is another type. And as you can see in this table below, they all have different proportions of different ions, different organic components, and nutrients for the cells. So this picture at the top right shows what mycoplasma contamination might look like. So comparing it with the picture that we saw earlier, we don't see as many cells sticking together, and we see them in these circular clumps, which are the bacteria.
With the HCT116 cells and lipopeptides covered, it is time to talk about our current progress and future plans. Currently, since our previously growing cells have died, we need to restart by thawing frozen cells that we can maintain and develop into a steady cell line. We have also learned RNA isolation on bacteria for future use. However, our next steps of research will require us to learn how to conduct MTT and apoptosis assays. For our lipopeptides, we have recently succeeded in creating a reaction by following our lipopeptide synthesis protocol. With evidence that our protocol works, we now need to synthesize lipopeptides that are up to measurement. Afterwards, we are to work on attaching the lipopeptides to HCT116 cells and see if they can induce apoptosis. These are our citations. And thank you.